Next talk is from Dr. Luis Montaner from the Winstar Institute in Philadelphia, and he's talking about boosting the natural function of the immune system during the HIV infection.
Bueno, buenas tardes y muchas gracias por esta invitación. Fue un placer estar aquí, pero voy a hablar en inglés porque, por el beneficio de todos ustedes, porque creo que los términos específicos no los sé muy bien en español, así que por ende voy a cambiar a inglés. So I'm going to have a discussion with you about the boosting the natural function of HIV within the context of a unique opportunity that we have to build a new team towards an HIV cure agenda. So we're going to discuss some basic overviews and assumptions of our cure effort in general, because I think it's important to review what is some of the driving paradigms that we think about when we think about pursuing a cure agenda, and some of the questions that are driving us to think about how to engage this question in a human setting. And then lastly, we're going to end by reviewing exactly what is our approach and new data to pursue a cure within the context of our ongoing work and expertise. So I think it's important to first remember that, you know, we do have an answer to stop the pandemic globally, and we have discussions of cure with a very specific objective of overcoming the infection, but when it comes to the global burden of HIV, antiretroviral therapy is effective, and we know that it can stop transmission. So I think from the point of a cure, we have to make a distinction between efforts that end the pandemic as we know it now and efforts that are targeted towards a cure. And when it comes to a cure discussion, there's a lot in the literature about how a cure is accessible or how a cure is not going to be possible. And you can find multiple discussions that have been coming out this year, but there are a couple of assumptions that we all seem to agree upon. The first one is that early antiretroviral therapy is going to be a beneficial strategy to limit the amount of the reservoir. And to some extent, the Mississippi baby was one case point. The Visconti cohorts in France are another example of individuals that have started early treatment. And there have been clinical trials that have tried to pursue this idea, specifically in Thailand, where they've treated individuals early. Interestingly, this year there were data from this cohort that showed that most of them actually have rebounded. So therefore, this idea that early treatment alone is going to be the answer for the human intervention as far as getting a suppression or a large number of people appears to be in flux. So I think that assumption, I think it's been challenged a bit since we first started to define this. However, when it comes to chronic infection, we know that we either have to get rid of the infected cell, deplete it somehow, flush it out because it's latent and quiescent. But regardless of which strategy we take, we all have the agreement that it's going to take some degree of immunological clearance to help whatever strategy we're pursuing. And when we think about a stepwise model, I think Nicholas had an excellent platform that I'm going to share, because it does sort of illustrate our thought process. You know, we obviously recognize that there is the control of HIV replication needs to be followed by ART, and that ART isn't complete. We still have RNA, we still have low copy number, so anything we can do to intensify suppression is a path forward. After that, we also know that we need to get after the infected cell and clear it. So any strategy that can augment this process is going to be considered beneficial. And at some point, whether that strategy is individually going to go after the latent cell or we need to reactivate it at the same time is another idea that is being pursued. And you can see how research could be segmented in each one of these components and be sort of cure-related research without necessarily being a strategy that takes you from beginning to end. However, as we're going to discuss, when it comes to interpreting whether what you're doing is beneficial, then here there's a lot of discussion, because it seems that the only strategy that we currently have to determine for certain that something is beneficial is by stopping antiretroviral therapy. Now, you could argue, does every cure study need to include a treatment interruption, or would you like to see an effect while individuals are on antiretroviral therapy first? And I think that's a dialogue that we currently are having in the field, because as you know or will review, measuring the virus in antiretrovirally treated people and interpreting what that means is somewhat of a challenge. So the leading questions, again, There have been reviews recently this year that I'm putting up here that you can sort of go for more specific discussion, but some of them are pretty dominant questions. Like, for example, why is the decay rate of the latent infected cell so slow relative to antiretroviral therapy? What type of virus persists if it's primarily defective? Which is the virus that we should be measuring relative to the persistence? And to what extent is a defective virus expression part of the immunodeficiency process of the virus to sort of get the immune system to pursue ineffective infections relative to the virus in itself? What should we measure when we think about cure-related strategies? Because we know that the total amount of DNA is not going to be completely effective viral expression. We know the RNA could potentially not be transcribed into protein. We know that once a protein is made, the particles are 99% effective in the circulation. So that when we have a dialogue about how we engage the cure question, we are faced with some of the limitations that we currently have, because even reactivated virus, even the quantitative overgrowth of virus is now also presenting challenges that it's somewhat restricted by how many times you activate it and what amount it truly reflects of the total latent pool. So that however way you discuss it, there's always going to be some qualitative note on the side that says, well, yes, but that may still not be the ultimate outcome in interpreting a cure-related strategy. Then he says, well, let's see what we're going to measure. I mean, we started the cure agenda with the issue that we needed to reactivate the latent cell, because if we reactivate the cell, we can actually pursue its demise by apoptosis. However, we know that T cells can survive, and most recently, we're now discussing the issue of clonal expansion as a persistence of the reservoir. So now we have the issue that we may actually have up to 3 different types of reservoirs that we're going to be evaluating when we think about a strategy and how to engage it. We've already discussed, very beautifully in the first discussion about the impact of tissue reservoirs and how each tissue may be presenting a differential retention mechanism for the virus relative to the cells that reside in each one of the tissues. Case in point, the lymph node and the follicular T cell being one primordial sort of objective of cure agenda, saying, well, show me a strategy that can reduce the lymph node burden in order for me to be perhaps a little bit more comfortable that a treatment interruption is going to have a consequential outcome if you pursue that. We've talked about the follicular dendritic cell as another one. Then when it comes to humans, I mean, this study sort of reflects the discussion that we've just had in the non-human primate, because this is a human study on autopsy patients where they've actually been able to show the detection of HIV DNA and RNA across multiple tissues. Again, raising this issue that an antiretrovirally treated, undetectable in infected individuals, you can find virus in multiple tissue compartments. So although we go after the blood, we can talk about the gut, we can talk about the lymph node. When you're thinking about reactivating the virus or where it may hide, I think there's a lot more that we need to be aware of as to where the virus may be. And that's perhaps a reason why treatment interruptions are so informative, because it doesn't matter where it is. If it doesn't come back, it's clinically significant. If you're trying to base the entire strategy on measurements that are indirectly made, you're always going to ask yourself, is that truly going to reflect the removal of the virus from a human infected person? Now, as far as the CNS, we've talked about whether it stays in the microglia, and I thought this work from Janice Clements was interesting. It's the first one I see where they try to reactivate virus directly from the CNS, and showing that an antiretrovirally treated macaques, they can actually show the reactivation coming from the CNS directly. And the question is, would a similar type of phenomenon be also present in humans? And that remains to be determined. So what started from a 1997 report of what was a latent, population that was remaining quiescent, I think now the discussion is shifting that this is a line that is stable, but it's not because it's static, because we may have expansion from one side and decay from the other, and as a consequence, you have a steady state. You could argue, well, why is the virus not increasing if it's due to a clonal expansion and there is no reactivation? I think that's a good question. We don't know the answer to that. And I think that whole dynamic of how we interpret the different mechanisms accounting for latency is a discussion that we're currently having. So I thought this paper was intriguing in that it says, well, since we don't really know what to measure, perhaps the time to rebound is the best measure of all. And they sort of presented a model as to how to interpret. And one of the things that was intriguing is they do note the SIV model is a lot more efficient in being reactivated, so that it may actually be a lower threshold, if you like, for control than the human system, meaning that if it works on the SIV, it's probably going to work on the human, because the SIV is more likely to rebound a lot quicker than the human. Now, with that, I think the data from the alpha-4 beta-7 showing that the remission of these macaques after treatment with this antibody strategy on top of antiretroviral therapy, and they have a large frequency of them remaining undetectable after this strategy, right here, almost like 80 days after, all the animals that actually did not respond against the antibody treatment itself become very intriguing, because it says, well, this is the only example I'm aware of a strategy on an infected animal that has been treated with antiretroviral therapy where the efficiency of remission was actually observed in almost all the animals. Because even the CMV vaccinated animals from Lewis Picker's cohort, it's 50%, and that 50% includes animals that actually blip along the way. So that's not 100% effective. We don't know the reason for the 50%, but that's not the case in this strategy. So I think that there is some hope, but there is a lot of uncertainty, because, for example, we don't really know what the mechanism for this outcome is. I'm just showing you that the measure of treatment interruption becomes very significant, because it's the best measure that we currently have. Obviously, the biology relative to the strategy targeting the MAD-CAM interaction in the gut highlights a very unique role for this particular recirculation in the context of the chronically infected antiretrovirally treated animal. That remains to be determined. But some of the correlates that were associated with this outcome were retinoic acid recovery, which would be an outcome of gut reconstitution. And interestingly, retinoic acid has been associated with reactivation of the latent reservoir as a strategy itself. And interferon alpha levels were actually also the second correlate of what these animals had that was distinctive about them. Now, within this context and this dialogue and this discussion, this is where we've sort of assembled a team to try to address the HIV cure agenda with all the caveats that we just discussed. And primarily, our focus is in trying to integrate different components of immunological strategies that could be used together, specifically highlighting those that we have preliminary data for already, meaning that we have trials where we have tested interferon alpha in humans, CCR5 sync nucleases, CD4 T-cell CARs, and broadly neutralizing antibodies. So we're trying to start from those strategies where we actually have some data in humans already, and then determine if we combine those, do we get any further ahead than where we have been. Obviously, with a heavy emphasis on trying to understand the persistent HIV, so that the two goals are determining the role of clonal expansion relative to the reservoir and its composition, trying to go beyond the blood, and also within which type of reservoir are we trying to measure. And second, the immunotherapy relative to strategies. The clonal expansion is primarily based on the observations from Bob Silicano, where he's shown the persistent clonal presence of a very specific blood-related representation of sequences that suggest one contribution of a clonal virus that is more efficient than others. And the whole discussion as to whether that reflects a preferential integration of some viruses that can actually sort of hijack the sort of maintenance of the T-cell repertoire in its homeostatic proliferation. And so there's a lot of pursuit saying, can we understand that those viruses that are within the homeostatic proliferation actually have landed in specific integration sites that allow them to do that? And if we were to identify these, would that be a way that we can then qualify the distribution of the reservoir in individuals on antiretroviral therapy, whether they're primarily in one particular type of cell or not, as far as the constitutive proliferation is concerned? And the interplay, if this is actually consequential, meaning can you actually show that this population resides replication-competent virus or not? Because as we just discussed, just because you can measure it doesn't mean that it's clinically significant. Now, the reason that this is important is that because when we think about strategies that our latency reversal, we have a population of latent viruses, and the idea is that as they get reactivated, we're going to thin out the population of viruses because of apoptosis and cell death. If clonal expansion and homeostatic proliferation is an adjunct to this process, then what we may be doing is actually redistributing the virus, because we may be proliferating a subset of cells at the same time that we're activating a parent population as well. Like, for example, take IL-15 or IL-7 or anything that would actually proliferate T cells. You could argue, which of these is going to win first? Is it going to be the immunological clearance and reactivation, or the clonal expansion of a latently infected population? And obviously, that remains a research question. Now, the area that we're particularly engaged in my laboratory is the issue of innate immunity, trying to exploit the antiviral mechanisms known to be associated with activation of type 1 interferons, which is a natural response of the host to a viral infection. And we're particularly focused on the type 1 interferons, which is with the particular emphasis on those that are therapeutically available, that were initially developed for hepatitis C treatment, in this case being primarily interferon alpha-2A and interferon alpha-2B. Now, the biology of these molecules interfaces the antigen-presenting cell as well as the natural killer cell and the B cell relative to ADCC. But I want to, perhaps if anyone's interested, we can talk about the differential between interferon alpha-2A and 2B and receptor interactions and T cell energy versus immunological clearance, because there's a lot of discussion about the fact that interferons can also be anergic in the context of chronic activation. But we're going to focus our discussion on antiretrovirally treated individuals, which is a completely different set point when you talk about interferon activation, because there's no antigen. So presumably, you're providing an interferon response in the absence of chronic activation. Now, if you're talking about HIV replication or SIV replication, then you're talking about predominance of both interferon alpha and interferon beta. And interferon beta has been shown to be preferentially associated with T cell energy in LCMV models. So I think we need to restrict our discussion and our interpretation of type 1 interferons specifically within the environment that we're going to study or discuss. There's no such thing as one soundbite that addresses the role of type 1 interferons across all sorts of biology, because I think that would be incorrect. So we've already talked earlier yesterday about multiple mechanisms of host intrinsic resistance, but the way that we like to think about it is more of an interface across multiple different arms of the immune system, trying to target both intrinsic, innate, and the interface with adaptive, specifically through antibody-dependent cytotoxicity. We've done a lot of studies trying to understand what correlates with control, and invariably the message that have come back from all of these correlative studies is the fact that those that control after treatment interruption, or viremic controllers, or elite controllers relative to KIR distribution and HLA expression, they all seem to associate with both an innate and a T cell response being concurrently present in the individuals. We've seen in elite controllers the fact that NK responses could be detected when T cells responses are not. But when it comes to treatment interruption, it's really the composite of both T cell responses and the preservation of the crosstalk with the PDC that remain correlates. So we move that further and say, well, to what extent is interferon alpha able to engage these, using interferon as almost like a replacement of the plasmacytoid dendritic cell, which makes interferon. To what extent can we use that to elicit effector mechanisms against HIV-infected cells? And invariably, we've been able to document that that's a very efficient mechanism by which to recruit the natural killer cell in detecting autologous infected targets. And we've pursued mechanisms for how this is happening relative to activating receptors that are required, giving us sort of like the basis to determine, well, then should we add this to an antiretrovirally treated person and see what happens. So we did that. We treated individuals in chronic infection with interferon alpha immunotherapy and stopped treatment to determine what would happen. We identified that about half of the people remained suppressed after stopping antiretroviral therapy, which was the first time that that was actually evidenced to be the case after an immunotherapeutic strategy. But most importantly, we saw this tendency of what was a slight drop in the but significant, of the integrated DNA levels in individuals that were controlling virus, which gave us the hypothesis that we perhaps were increasing clearance of infected cells, whether they're replication competent or not, relative to the presence of interferon. When we look at gene expression that was associated with the drop of the integrated DNA, all the genes that were associated with the drop were related to T cell natural killer cell activation and killing mechanisms. Again, giving us the inference that the mechanisms associated with the beneficial response to the interferon were actually immune activating as predicted by the in vitro data. This is after the data we had after like 5 weeks of interferon. This is in the open-ended interruption. We had less observations, but the theme is very similar to the one above. Now, the question is, that was with interferon alpha 2A. So we then did a second study with interferon alpha 2B, adding here 20 weeks of interferon, and again with a 4-week interruption here right in the middle. Now, the previous study was primarily looking at blood levels, so that we wanted to also explore the rectal compartment, because as you just discussed, getting a sense of what's happening in the tissue is an important parameter to determine whether what you're doing is perhaps consequential. It's still not perfect. I think we would have liked to get lymph nodes at the same time, but the patients and the providers are saying, well, you get lymph nodes, you get no patients. So you can either do blood and rectal, or you don't do the study. So we elected to do the rectal on everyone, and with the understanding that the rectal was going to give us a window on RNA and DNA, that the PBMC was going to be a lot more challenging to evidence. This is RNA and DNA coming from all the different GI compartments, and you can see that the rectal tissue is pretty representative of the entire spectrum, and it's a lot more convenient as an access from a patient. So we decided to obtain the rectal biopsies. Now, before we interpret, remember, we're shifting from 2A to 2B. So one of the first things we wanted to do is, during that 4-week interruption, is the suppression frequency that we observe with 2A similar in the 2B treated, because they're different drugs. So this study is showing you those that were suppressed after 4 weeks of interferon alpha 2B as compared to the parent study of interferon alpha 2A, and they're basically comparable. It's about 45 to 50%, which is exactly the same frequency that we saw before. Now, in this case, we had the rectal biopsies at baseline, and what was surprising, at least to me, was the fact that detection of RNA-positive hybridization when you use the entire HIV sequence to hybridize was actually not that hard to find in more than half of the patients that we studied. It wasn't present on all of them, but it was certainly detectable in a large frequency of them, and this is sort of the signal that we get. This was done by Ying-Seng Lee over at University of Nebraska, which was, he's coming from Ashley Hass's group, so that he can identify the cells and the area relative to the detection of the signal. So this is, again, this is PIP3, this is patient 3, baseline, visit 2 in the study, and you can see that this signal is coming from very much next to the epithelium. Interestingly, it wasn't as evident in the lymphoid nodule that was rich with CD4, and it's actually CD4, CD68 cells that are around this area. But after interferon in this particular patient, we couldn't detect it. This is the same, this is visit 9, the same individual, and we can't detect the presence of the RNA. Now, when you take all the patients that had detectable levels at baseline, this is the image that you get, meaning that invariably for most patients, you see a very uniform decrease of the signal coming at the second time point. And we did this twice. We actually send them blinded and non-blinded. We send them in 2 different batches because it was like, we were trying to make sure that this was actually reproducible, at least within the context of our own study. And invariably, the same data kept coming back. So now, in this study, we had leukapheresis so that we actually could recruit different teams to do different type of assays, because as I noted, you can have a whole repertoire of assays that measure different amounts of HIV, and the challenge is, how do you interpret them? So we had Nicolas Chamont in Canada doing integrated DNA, Doug Richmond doing total DNA over in California. We had Merck, Bonnie Howell doing tilde assays relative to the beginning and end, and it was by design that we were trying to farm out measurements to determine to what extent are common themes coming back from the different types of analysis. Now, you can do correlations at baseline of all these different variables when patients are on antiretroviral treatment before they start interferon. And these are the correlations. You can see that the total DNA from Richmond is correlating with Schumann's integrated DNA, which is something perhaps one would expect. The P24 assay on the tilde the first day with the ultrasensitive P24 is correlating with the RNA measurement of the tilde. The tilde measurement in RNA is correlating with the integrated DNA from Schumann, and the two DNA variables are correlating each other. What was interesting is that the rectal RNAs weren't really correlating with anything in the peripheral blood, suggesting some degree of compartmentalization or differential relationship between what's actually driving the signal of the RNA in the GI and what you can actually measure in the blood. Now, I don't know if you had the opportunity to do this sort of thing in the non-human primate and to whether that's real or not, but that was our observation. Now, when you look at all the individuals beginning and end across all these different variables, we already addressed this phenomenon here, which is what I showed you. The first study had a significant drop in integrated. This is 0.07, and you can see that it's actually not that remarkable, at least the difference between the two time points. And all the other time points are not showing a significant difference. Now, one interpretation of this is that there's not much happening, meaning that interferon 2A dropped the RNA, but it's really not having any impact on the other measures, apart from a couple of individuals, like this one, for example, right? So we looked at the data in a different way. We plotted the level at baseline, meaning what they actually presented when they presented to the trial, to the delta change that was actually detected after the interferon. And what you can see is that those individuals with the highest signal were the ones that had the highest drop, whereas those that were very limited at the age of the signal, you couldn't actually hardly see anything. Now, if you replot all the variables relative to that particular strategy, then you can see the RNA here, but now you can see the tilde and the 2LTR, P24, sort of kind of like a common pattern that suggests that perhaps, and I'd say perhaps, there is an effect of interferon, but it's actually being restricted in relation to the baseline variables that people enter into the trial. Because remember, when we recruit, we say, can you consent and take interferon? And if you say yes, we will study you. We don't actually first measure the tilde and the integrated and all these measurements and only choose those that have higher levels. Now, the argument against this is something called regression to the mean, which means that you have a distribution in a bell curve, and if you measure the end of the distribution, the probability is that if you measure it again, it's going to be in the middle of the distribution. So that for that, you would need to have multiple measurements, or you would actually have to have a control arm as opposed to a one-arm study. Now, I don't think that this could be explained by regression to the mean, but that's a possibility. But never the case, we've already gone through most of these, so I'm not going to repeat it. We have this study that's actually ending recruitment now. It's about 54 individuals, 18 per arm, and this is we're about 42 of the 54 recruiting. But the important thing is that this is a randomized trial now where we're actually testing the effect of the interruption, the continuous interferon, and most importantly, an ART-only arm. So that if indeed there is an effect, as I would hope there is, then it should be confirmed by this particular arm, because that obviously would be the appropriate control to have. In addition, we'll do multiple repeats as well. Now, the direction that we're taking with the new grant is actually complementing the interferon with broadly neutralizing antibodies, because in vivo, we already know that broadly neutralizing antibodies can have an effect, at least in antiviral pressure. But in vitro, if you take an autologous system where you take the blood, you isolate, you infect, and you take the NKs of the same person a week later and you put them together, you can actually evidence that in infected targets, which are here, the percent of lysis in the presence of interferon can be augmented, but in the presence of interferon and broadly and antibodies, you can actually raise it even further. And that's true in isolates that decrease MHC class 1 very efficiently, like this one, or isolates that don't decrease MHC class 1 very efficiently, which the direct cytotoxicity is not as high, suggesting that interferon could give you an additive effect of direct killing plus ADCC or ADCC relative to the MHC class 1 downregulation of the virus. Now, that would predict that the combination of broadly neutralizing antibodies and interferon should be more efficient than interferon alone. So the trial design is as you would expect. You add them both together, you stop ART, and then after that, for the first time, we'll have an open-ended interruption, which we've never had the opportunity to do before. But now that the winds have changed, so the FDA and the regulatory environment in the US is a little bit more accepting of stopping everything and seeing what happens, which I would say about 8 years ago, this was, because of the STEP trials and others, this was not possible. So we will identify what correlates with this response. We will do the clinical trial, and then there is a component with David Weiner about adoptively transferring antibodies by DNA vectors, taking advantage of his DNA vaccine technology. Now, the team also has a T-cell response angle, which is based on the delta, on the data from Pablo Tebas and Carl June on the administration of a delta CCR5 zinc nuclease to protect the CD4 cell. And this is the data that they showed, where they showed this was safe and tolerable, and they interpret that this individual here had an antiviral response as a retention of the vector, and as a consequence, it suggests that this could be an antiviral strategy, at least from the point of view of protecting the CD4. So that we intend to continue to use this strategy, at least to protect the cell, because of the long-term evidence of its use, its safety, and the fact that it is retained. Now, that's probably not enough, as you probably saw the treatment interruption, so that what is being proposed is actually to join the the zinc nuclease with the CD4 T-cell CAR. And the reason for that is that the CD4 is a conserved epitope to the envelope interaction, so the virus can't really mutate away from binding CD4. So if you have a T-cell that has a CD4 molecule tied to a CD3 zeta and a killing mechanism, that would make this interaction result in a killing interaction against the viral infected target. Now, this was tested in HIV-infected people before, but actually had hardly any antiviral activity, even though it was retained. But the reason that they interpret that it didn't have antiviral activity is that the CD4 would activate the CD8, which would then express CCR5, and as a consequence, the CD8 would be infected and wouldn't necessarily affect killing. So the hypothesis now is that if you actually put the CD4 and you protect the cell with a CCR5 zinc nuclease, this killer would be retained and would actually be able to affect an antiviral strategy. So that's testable. So that's actually the trial that will be moving forward from the gene therapy side of creating a CD4 and a CD8 cell, because you would actually deliver the vector to both, that could have the killing potential in combination with a protection from infection at the same time. So the trial design is basically you administer the gene therapy, half of the individuals will get reactivated with, say, panobinostat or an HDAC inhibitor, and then you stop ART and you determine whether that is able to mediate suppression and to what extent is it long-term afterwards. Now, all of this is kind of like consistent with at least the paradigm that we're trying to develop strategies that will reduce the amount of virus by at least interferon and broadly neutralizing, but it's more as a prelude of what may be a secondary strategy that will sustain suppression. So neither of these are thought to be the end result, but just tools that we expect to join with other strategies. Now, if there are other ideas, like for example, alpha-4 beta-7 or any other strategy, you could start to sort of mix and match about what combination would be most efficient. But our understanding is that to get to a functional cure or to an ultimate sterilizing cure, you're probably going to have to use combination strategies anyway. So we might as well start to see what we can learn from combining those that we have data for. So with that, thank you very much, and I'll take any questions.
We are a little bit late on time, but I think we can have a couple of questions.
Luis, that was lovely. The question I have for you is the timing of these interventions relative to antiretroviral therapy initiation. There's some discussion that, or some thought that the patient needs to be completely suppressed before you have these intervention strategies, and then there's also a contra thought that the active reservoir, the cells that are actively replicating at the time that antiretrovirals that are on board, are important for that seeding of those long-term reservoirs. What are your thoughts on the timing of these interventions?
I think those are important components to refining any strategy that actually meets the first goals, but for a lot of these strategies, we're going after the best patients, those with high CD4 above 350 or 400, high nadir above 200, long-term suppression, at least a year of undetectable. Most of them are under 70 or 65 years of age, so we don't get, sort of into other types of biology. So that a lot of these initial strategies are targeted to what would be the best case scenario. Now, the idea is that if you find an effect in these individuals, then you can start refining to what extent can you extrapolate time to therapy. But the idea is that we're still going after what represents the majority of people that you're trying to target, which would be those chronically infected long-term ART, as opposed to having started ART very early. I think those are important because they're mechanistically going to give us insights as to what may be preferentially going to work. But logistically speaking, they still represent a very limited subset of people that the strategies that would work on them would then need to be shown that it works on everyone else. So it's kind of like the same argument.
Yes, thank you for the talk. It was great. I have a question regarding it looks that interferon can be of benefit in some situations, but the chronic stimulation of interferon could not be as good. So my question is, are you looking at biomarkers of the good or bad response? I mean, some ISGs.
Absolutely. I think we're currently doing that. In this study, in the interferon alpha 2B study, the second one, we were able to, in the first study that we did, interferon alpha 2A, we hypothesized that if we actually looked at the ex vivo response to interferon alpha per the intracellular staining of STAT1, that we were going to qualify which patients were still responsive to interferon. We found that everyone that was getting interferon dropped their STAT1 phosphorylation response ex vivo uniformly, even though if you stimulate it with interferon gamma and you gate it on the monocyte, the STAT1 was not decreased at all. So it was very specific to the interferon alpha receptor. Now, I don't know if you've done intracellular staining for for these types of STAT1, but you lose a lot of cells. So it becomes kind of like, you can get a result, but it's suggestive because the method is very challenging to execute. So in the second study, rather than STAT1 intracellular, what we did is we did the same ex vivo exposure, but this time looking at gene expression, because the hypothesis would be that if STAT1 is decreased, ISGs will be decreased. Now, in addition, the hepatitis C field has identified multiple molecules that are associated with the interferon refractory state. And these are USP18, ISG15, there's a couple of other ones. So that one hypothesis is that the refractory response is going to be inversely related to the retention of the antiviral effect, and that in turn is related to the ISG induction of genes. Now, so that's currently the hypothesis we're trying to explore.
So just a quick last question. So it's very interesting work. Congratulations about the role of interferon. So what about you have a talk about the exogenous administration of interferon. What about the manipulation of the PDC and trying to regulate the PDC to produce interferon? And do you think this is important, and do you think this is feasible, and do you think how can we do it if you think is...
Yeah, I think that the stimulation of the PDC has been a particular target for, say, the TLR7 agonist, which Gilead is developing a TLR7 agonist based on the activity of the PDC in activating the reactivation of the latent reservoir. So interestingly, in that system, interferon receptor, if blocked, can actually block the reactivation in vitro of the TLR7 agonist response. Now, if they add the interferon to the same assays, they don't see the same effect of the reactivation as targeting the PDC through the TLR7. So I think that when it comes to the therapeutic intervention. I think the recombinant interferon is probably an easier system to interpret and predict. When it gets to a more of a cellular response, then I think that you engage a much broader mechanism of action that perhaps is more difficult to interpret when it goes one direction or another, because there's going to be a lot more involved than one particular molecule being produced. So I think the PDC are useful in assessing the biology and perhaps in the TLR7 agonist, but from a point of view of an antiviral strategy itself, I think that I would still favor the interferon side, specifically because we haven't talked about beta. But beta engages the receptor to an extent that it energizes a response, whereas alpha doesn't. And I think that in the context of a LPS response or a TLR7, you're going to get alpha and you're going to get beta, and the whole dynamic is going to be very different, at least from the point of view of the interferon receptor.
Okay, thank you very much.